Swayam Prabha. Digital India. Educated India.
Good morning, everyone. I'm Chandra. I'm a faculty here in the chemistry department. I'm an assistant professor here. So, my research interest is basically in the area of biophysical chemistry, or to call it more molecular biophysics. Okay, I try to understand the physical chemistry properties of biological systems. So, more complex systems than a simple chemical system. So, we try to understand the kinetics point of view, thermodynamics point of view, and all mechanics point of view. All these properties of biological systems, mainly proteins, enzymes, and nucleic acids, and these things, which are nothing but chemical entities, but more complex systems than simple chemical systems. So, today I am not going to talk about anything about the research. Today I am going to talk about a simpler topic which is core to the understanding of kinetics of enzymes, which is called enzyme kinetics. So, how many of you have read or gone through or have had a course on enzyme kinetics? None of you read anything about enzyme kinetics? Can you show me by the raise of hands so that I know? I am not going to ask questions, so don't worry about it. I know that once I say this thing, then people are referred. So, okay. So, I can say at least 5, 6 people or 7 people. So, don't worry, I am not going to ask any questions on that. But so, you have heard of the word enzyme kinetics, right? And you have heard of understanding enzyme. So, you all know what is kinetics, right? What is kinetics? Is it just rate of reaction? Studying the rates of reaction. But what information does kinetics give you? It gives you how fast the reaction goes, right? And what else it gives you? What are the factors affecting the rate of the reaction, like temperature, pressure, concentrations, and all those things, okay? And what else? Order of a reaction, very good. So, you can also calculate the rates of order of the reactions from the rate of a reaction, right? So, basically by knowing the kinetics, if you understand the entire kinetics of it, you can calculate the order of the reaction. How do you calculate the order of a reaction from understanding the kinetics? What is order of a reaction? This is something very basic, right? What is an order of a reaction? So, there is a dependence of a rate of a reaction on the substrate concentration, right. So, either it is linearly, so you have a rate of a reaction, right. It is either proportional to substrate concentration, or it is proportional to your substrate square, or sometimes it might be independent of the substrate concentration, right, per zero. That means it is a constant value. So, so it could be independent of it, right. So, you can have a rate of a reaction basically by understanding the change in the rate of a reaction with the substrate concentration. You can calculate the order of the reaction. Now, what is the use of determining the order of a reaction? What is the information that you get by determining the rate of reaction or order of a reaction? Is order of a reaction, can you calculate the order of the reaction by looking at a reaction? You must have heard of two terms, right? Order and molecularity. One is molecularity, the other is order. So, can you determine the order of a reaction just by looking at the reaction? Order of the reaction cannot be determined by just by looking at the reaction. Only for simple elementary reactions you can do it, but order of the reaction needs experimental validation. So, you do an experiment, you determine the rate. What is rate? What is rate? Speed of the reaction, but how do you determine the rate? Particular time? So, you are monitoring the change in the concentration of the reactant or change in the concentration of product with time, right. If you want to write it mathematically, you can write minus substrate, have a reactant as R minus dR by dt, or it is, if it is a product, it could be dP by dt, right? So you are trying to calculate the change in the concentration of a reactant or change in the concentration of product with time. Now you can ask me, how do I calculate the change in concentrations? No, how do you calculate? How do you, so when you are doing a reaction, how do you determine the change in concentrations of either a reactant or product? Have you done any of practical experiments in your first year MSc or even +2, right? How do you determine the concentrations? Now if I have a reactant A going to B, right? Now I need to either monitor A or monitor B. So my rate of the reaction is either minus dA by dt or dB by dt, right? So I need to monitor either this or that. So I have to monitor either the change in concentration of A or change in concentration of B. So in a normal reaction, when you are doing it in a lab, how do you determine the change in concentrations? So how do you know whether a reactant is decreasing or a product is increasing? How do you determine that when you are doing a reaction? You by visually, suppose everything will be mostly reactions are transparent, right? If there are transparent reactions by just selling, you cannot say how much reaction is going, right? You have to do some kind of analysis on that, right? So what kind of methods are you aware of? Titration methods, for if it's an acid based titration, or based on that you can determine, yes. Conductometry you can do, gravimetry, potentiometry, or you can even some of the compounds also absorb, right? You can determine absorbance, or if it's a simple organic molecules, you can do a TLC reaction. You can just take the reaction mixture and put it on the TLC. You are aware of TLC, right? So you can and see there will be a substrate point or a mark and then a reactant. So you can determine the change in the concentration. So there are many methods, but let us not worry about the methods. But you can determine the rate of a reaction by monitoring the, so at a different time points. So at every different time point, you measure either the concentration of reactant or the concentration of product, and you can determine the rate of the reaction. Now if you actually determine how the rate of the reaction is changing with the concentration of the substrate, you can determine the order of the reaction. Suppose now rate means what? Change in concentration. Suppose I have, to start with, I have 10 molar. After so to start with at 0 minutes, I have suppose 10 molar, and after 5 minutes I have only 8 molar. What is the rate of a reaction? What is the rate of reaction? When I have it, basically you can do it for different time points, right? So this concentration 10 minus 8 by 5 minus 0, right? So 2 by 5, right? 2 by 5 molar per minute, and then you can convert into seconds or something, whatever you. So you can determine the rate. Now you can determine this rate of the reaction at different concentrations of substrate. That means you can change the initial concentration of A. You can have 10 molar. Again, you can start it with 8 molar. You can start it with 6 molar. You can start it with 4 molar. So this rate, this initial concentration can be changed, and you can determine the rate of the reaction. Now if the rate of the reaction is independent of the substrate concentration, this is called a zero order reaction. That means your R rate of a reaction is independent of the substrate concentration, right? It's a constant value, right? S bar 0 is 1, so it's independent of the substrate concentration. This is called rate constant. Now if the rate of the reaction, suppose is increasing with the substrate concentration linearly, what will be the order of a reaction? First order reaction. R is proportional to S, right? Substrate. Now you can have a second order reaction. What will be R proportional to S? So what will be, it will be R per S square. So it will increase, basically, or not this, but it will have a, it will increase it as a square of the function, right? It will increase very quickly. So you can have rate of the reaction. But what information do you get by determining the order of the reaction? So order of the reaction is only determined experimentally. You cannot, just by looking at the reaction, you cannot determine the order of a reaction. Molecularity you can determine, but order of a reaction can never be done. So what information can you get, but what is the use of determining the order of a reaction? Yes, what kind of characterizations? So you can actually predict or you can hypothesize the mechanism of the reaction. If there are intermediates, you can also validate those intermediates, right? So you can determine the mechanism of the reaction, not actually determine, but you can validate it, okay, or predict a mechanism of the reaction, or you can also see if validate any intermediates are present, those can be done. And you can also, by determining the order of a reaction or rate of a reaction, you can control the how fast or how slow it goes, okay. Now this is something that's basic, in the sense there are multiple uses of determining order of reaction, and some of them is this. Now, but coming back to this, the topic that we started, what we talked about is only kinetics part of it till now, right? But that is not the topic that I am going to talk about. I am going to talk about enzyme kinetics. So why do you think enzyme kinetics is important? Some of you have read about it, right? Why do you think enzyme kinetics is important, and how do you think it will make that it has the understanding of enzyme kinetics started long ago, more than like 70 years ago, but still it is a hot topic that still people try to understand enzyme kinetics. Why do you think enzyme kinetics is important or different from a normal chemical reaction kinetics? Okay, very good. So enzyme kinetics are basically, enzymes are biological molecules. So it happens in the biological system. So you, by understanding the enzyme kinetics, you try to understand the reactions that are happening in our body or in any living organism. Okay, that is a biological understanding. From kinetics, from chemical point of view or mathematics point of view, is there anything interesting? We will come to that. But first let me see, how many of you know what are enzymes? How many of you know what are enzymes? Have you heard of the word enzyme? Okay, what are enzymes? Biocatalysts. Okay, I'll just write down so that I hear whatever I hear from the people. So some of it are calling it as biocatalysts. Okay, and anything that you know about enzymes, they are biocatalysts. Okay, huh? They are largely made of proteins. So most of the enzymes, or the large portion of the enzymes, are proteins, but there are also some carbohydrate based glycoproteins, other things, but still most of the enzymes are proteins. Okay, and what else you know about enzymes? You all know what are proteins, right? I don't have to tell you what are proteins, right? Proteins are polyamino acids, right? So polypeptides basically, right? So why do you think enzyme kinetics is important? So if you take a normal, so the first thing is, let us take it as a biocatalyst. Okay. Now a normal catalyst, what will a catalyst do? Alter the rate of the reaction. Okay, when we talk about specifically catalyst, we are talking about something that increases the rate of the reaction. There could be something that decreases, which we call inhibitors or poison or something like that, but generally catalyst is used for something that increases the rate of the reaction. So biological molecules or proteins that are increasing the rate of the reaction, these are called enzymes, right? One of the key features of enzymes is one is highly selective or specific, and two, they accelerate the reaction. So if you compare a normal catalyst with the biocatalyst, which is enzyme, right? Enzymes are very specific. That means if you have a specific substrate molecules, the enzymes are not, when they have a set of substrates, they are only even if you give million other compounds, they are going to only interact with a specific substrate molecules. So they are very selective. This kind of selectivity you generally don't find much in a lot of chemical catalysts. So you can say that this is the epitome of specificity. Enzymes are highly specific in what they are looking at, and the catalysis rate is also much more than what a normal catalyst can do. So the rate means the enhancement. Catalyst will do what? Catalyst will enhance the rate of a reaction. Okay. But for a normal chemical reaction, if you have an uncatalyzed reaction, A going to B, so if you have a rate of a reaction versus substrate concentration, Suppose it's a first order reaction. How will my plot look like? Linear, right? So for an uncatalyzed reaction, you have, so basically for an uncatalyzed reaction, you have for entire reaction, for A going to B, this will have only one order, right? So it's either if it's a first order, it will be like this. If it's a zero order, it will be like this. If it's a second order, it will have a second. So for one reaction, if it is uncatalyzed, it will only have a single order, right? But for enzyme catalyzed reactions, you could have multiple orders in the same reaction, for the same reaction depending on the substrate concentration. For example, a simple Michaelis-Menten kind of behavior, if you see. Okay, so this is the same, I'll come to the derivation, I'll show you where this expression comes from. But for the same reaction A goes to B, if it is an enzyme catalyzed reaction, if it's an enzyme catalyzed reaction, depending on the substrate concentration, you can have different orders. That means here you see it's a first order reaction, it's a linear, then it is some kind of a curvy nature, it is a fractional order, and here you see it is independent of it, so it is a zero order. So that means for the same reaction A goes to B, depending on the substrate concentration, the reaction can go from a first order, a fractional order, or a zero order. So this is a specialty of a mathematical specialty, or this is the specialty of an enzyme catalyzed reaction, that for the same reaction, it can, depending on the substrate concentration, the order of the reaction can change within the same reaction. So that means you can fine tune the rate of the reaction by changing the substrate concentration, because when you change the order of the reaction, you can fine tune the reaction rate, right? So you can fine tune the reaction rate by changing the substrate concentration. So this is something that you don't find in any uncatalyzed reaction. Only in, so this is one of the Michaelis-Menten catalyzed reaction. There are other kind of reactions like which have a sigmoidal curve and other sigmoidal nature and other things that we'll not talk about it now. But you need to understand that an enzyme catalyzed reaction is interesting to understand, because first thing is, many of the reactions, chemical reactions that happen in your body are done by enzymes, right? Or you can say most of the chemical reactions that happen in your body, or all the chemical reactions that happen in your body, are done by enzymes. So it is important to understand how these catalysts are altering the reaction rates inside the body. So they have a lot of biological significance, right? Second one is, if you want to understand, so enzymes are not just altering the rate of the reactions, but they are very specific. If you have looked at the catalyst, a normal metal catalyst or either nano catalyst, other kind of things, compared to any of this, the specificity, that means if I have to, if only this reaction has to undergo, if my substrate is only X, it will only interact with X. No matter if there are hundreds of different molecules, only if I have to interact with X, I'll only interact with X. That means it has a very high specificity. That means so in a body like ours, where there are some millions of chemical molecules, so it is not like your RB flask or reaction vessel where you are putting in a specific reactant, right? You are putting in a specific reactant and you want a specific product. But in your body, that is not the case. There are multiple or millions of chemicals, but only one specific reactant has to be converted into product, right? So for that, a normal catalyst will not do. Only an enzyme can do that because of its high specificity. And then the other thing is, it acts, as I already said, any catalyst will accelerate the reaction, but enzymes accelerate the reactions approximately. So are you aware of any other catalyst generally? How many times does a catalyst generally accelerate the reaction? Depends on the catalyst, but what are the numbers that you are aware of? A catalyst can increase the rate of reaction 10 times, a catalyst can enhance the reaction rate by 100 times or something, but enzymes generally enhance the reaction rate by 10 power 6 to 10 power 9 times. That means a million times to 10 power 9 times. That means if the same reaction you want to do it in a flask, in a RB flask in a lab, if suppose it takes 1 second for that reaction, an enzyme can do it in a microsecond. That means it enhances the rate of the reaction so many times with high specificity. right. So that is, and mathematically what is interesting is any uncatalyzed reaction, generally for a specific reaction the order of the reaction is constant, right, throughout the reaction. It is not, depending on what kind of order it is, the rate of the reaction, the substrate concentration will have a specific nature. But enzyme catalyzed reactions can have different orders across different substrate concentration. So you can modulate the reaction rates by changing the substrate concentration. So a reaction, the order of the reaction here is different than here is different than here is different, right. So that is why it is very interesting to, although enzymes are biological in nature, you can understand the kinetics of these enzymes or the behavior of these enzymes by studying them in the lab. But even then there is a huge specificity for this. Okay, so we will talk about how these enzymes actually, what is the speciality of this enzyme kinetics further. Okay, so is it clear why we should study enzyme kinetics? Is it clear why is it important to understand the kinetic? Because it's not just for understanding. If you look at a lot of drugs these days, any of the medicines, many of the, if you are taking a medicine for blood pressure, suppose someone in your family might be taking a medicine for blood pressure, right? What they will be doing, they will be taking a molecule which basically reduces the ACE receptors. Okay, so that is nothing but it is basically reducing the activity of specific enzymes. So that means reducing the activity means it is reducing the kinetics of that, right. So it's not just about understanding. Even if you want to design a proper drugs, right, if you want to so suppose you see the kinetics of various drugs. Some drugs you take every 3 hours, some drugs you take every 12 hours, some drugs you take every 6 hours. So you need to determine how efficient is drug, right. So the drug efficiency is also basically coming from your, because that you are basically inhibiting an enzyme or activating an enzyme, right. So you are basically determining the rate of those reactions and you are doing it. So for that you need to understand what is enzyme kinetics. So not only from a fundamental point of view it is very important because of these reasons, but even application point of view, if you want to develop a drug, most of the drugs these days are actually inhibitors of the enzymes, or they either inhibit or activate the enzymes. For example, cancer drugs, there are a lot of cancer drugs, right. Cancer drugs are nothing but there is an enzyme called kinase. Okay, that enzymes that inhibit kinases are kind of cancer drugs. So all this, so for that you need to understand the mechanism of the drug action, you need to understand the kinetics of the drug release and kinetics of drug inhibition and all this. So for this first you have to understand how fast is that enzyme, only then you can try to modify those enzyme reactions, right. So not only from the fundamental point of view, from understanding or from designing the drugs, for determining the efficiency of the drugs, for understanding how often you should give a drug, right. For all these things you need to understand enzyme kinetics. Okay, so we will talk about the applications if we have time, but we will see why this enzyme kinetics. So till now we have seen why the enzyme kinetics is important, and let us see how is enzyme kinetics different from a normal simple chemical kinetics. Okay. Now this is a suppose the catalyst is there. Now I have a delta G or a free energy. Okay, I have my reaction coordinate. So okay, you all have seen this kind of free energy versus reaction coordinates diagrams, right? Now how do you think an enzyme will change the free energy profile of this? What will enzyme do to this? So this is a free energy of the reaction. If I have a reaction A going to B will have a free energy, right? Gibbs free energy. So if you plot the Gibbs free energy versus reaction coordinate, suppose it looks something like this. Now how will an enzyme modulate or change this Gibbs free energy? Reduce the activation energy. Okay, and does it also modify the initial energies and the final energies? This is suppose reactant, this is product, this is your activation energy. So somebody said it will reduce the activation energy. Does it also modify R and P, the energies of R and P? So definitely it will reduce the activation energy, but so is it going to be like this, or is it going to be or something like it is going to be which one will it be? Yellow one? Yellow one means it is reducing the energy of reactant and product also. Orange one means it is keeping the reactant constant, increasing the energy of product. You are telling all the possible colors. Yellow is over, orange is over, then only white is remaining. Two whites are there. Which white? Yes. So basically, a catalyst will not alter the reaction or product. It will only change the activation energy. A catalyst can never alter or cannot change this or this. That means if a reaction is non-spontaneous, so generally when do you call a reaction spontaneous? When delta G is negative, right? That is when you call it a spontaneous reaction or when the reaction is feasible. If a reaction is non-spontaneous, no matter what catalyst you use, the reaction will not happen. So it is not going to change or alter this energy difference. It is not going to change the energy difference of the reactant or product. It is only going to change the activation energy barrier, that's all. That means if a reaction is, so that's right. So the catalyst doesn't alter the thermodynamics of a reaction, it only changes the kinetics of a reaction. That means it only changes the activation energy barrier. That means you have R and P here, it will only reduce this, but it is not going to affect the R and P energies. So suppose if you have a reaction, delta G is positive, no matter how efficient your catalyst is, this reaction is not going to happen. Only if delta G is negative, the spontaneous reaction, for that the rate of the reaction can be changed. So that is something that you should keep in mind. So let us take one quick example to for you to appreciate the enzymes. Okay, then we will come back to the mathematics for the chemistry part of it. But I'll just quickly show you one example of. So by the way, are you aware of any enzymes? Yes, name few. Zymase, okay. Huh? Invertase. What does invertase do? Glucose to sucrose or sucrose to glucose? It converts sucrose to glucose and fructose. Okay, and urease. And what else are you aware of? Lactase. So do you know what are ACE? All these have ACE. Any enzyme will have ACE. All the enzymes will have ACE. Any enzyme that does hydrolysis reaction, right, that will only have ACE. ACE is generally something that is related to hydrolysis reaction. So if you see peptidase means something that hydrolyzes the peptide bond, right? Invertase, that hydrolyzes your sucrose into glucose and fructose. And like there are other enzymes also, like you have heard of insulin? Insulin is also an enzyme, right? And then you have many other, like some of the most, I'll show you one enzyme called carbonic anhydrase. Okay, have you heard of carbonic anhydrase? Yes, okay, so what does carbonic anhydrase do? Carbonic anhydrase. Carbon dioxide to carbonic acid. So it can do a reversible reaction of converting carbon dioxide to carbonic acid. So right. So this is a conversion of carbon dioxide to carbonic acid. Is this an important reaction? Is it an important reaction? In our blood, yeah. So either you take a respiration process or you take a photosynthesis like a process, any of those processes where we inhale, there's a lot of carbon dioxide evolved during this, right? That has to be for maintaining the electrical, for maintaining the pH neutrality or something, it has to convert into carbonic acid. Even in photosynthesis where the plants take a lot of carbon dioxide. So this is a very important reaction in the body that is actually monitored by carbonic anhydrase. Is this a spontaneous reaction? CO3 plus H2O gives HCO3 minus plus H plus. It's a spontaneous reaction. If you want to do it in a test tube or if you want to do it, the general time scale is 10 power minus 1 per second. Okay. So that it is generally in the scale, time scale of 10 power minus 1 per second, the rate of the reaction. But if you do it so slowly in the body, that means you are not taking like it is only convert. So if you are basically for convert, so how much time does it take to convert one carbon dioxide to carbonic acid? If it's 10 power minus 1 per second, how many molecule, how much time does it take? So it takes 0.1 second, right? No, it takes 100 seconds. It takes 100 seconds, right? 10 power minus 1 per second. So 10 power minus 1 molecules are converted into this, right? So it takes 100 seconds to convert this one. So it's a very slow reaction compared to what happens in our body. So in our body, carbonic anhydrase does it at 10 power, the rate of the reaction is 10 power minus 7. It increases to 10 power 7 per second. It enhances it around 8 times, actually 10 power 8 times, close to 10 power 8 to 10 power 7 times, depending on what you are dealing with. Now let us look at quickly the conversion of this. Okay, so for those of you who are new to the system of enzymes, so this is how the chemical nature of enzymes is. So you have polypeptides, okay, you have amino acids that are there, right? So you have amino acids, then they form polypeptides, then they take what is called as secondary structure. Proteins fold into secondary structures, and these secondary structures further fold into what is called tertiary. So although we are writing it as cartoon, all these are nothing but your peptide bonds, okay, with the side chain amino acids. So you have secondary structure which fold into tertiary structure, and these tertiary structures assemble to form a lot of these quaternary structures, or generally which we call it as enzymes. Most of the enzymes are multimeric protein assemblies. So this is how the enzymes are actually. So this is not some cartoon or something. These are nothing but this is a cartoon representation of these peptides. So if you see this R, these are not simple alkyl chains. These are there are 20 essential amino acids, right? So depending on the side chain of amino acids, you will have a various R in this. So they will depend. So most of this is governed by non-covalent interactions. So the folding of a protein is governed by non-covalent interactions. So can you name few non-covalent interactions? Hydrogen bonding, van der Waals forces, hydrogen bonding, yes. Electrostatic interactions, which are called salt bridges, okay, and then hydrophobic interactions. Okay, hydrophobic interaction. So none of these folding or none of these things happen because of or the covalent bonds do not govern them. It is mostly the non-covalent bonds that govern this and form. So this is how a reaction happens. Okay. Now let me go to the example of carbonic anhydrase. Okay. So if you look at a carbonic anhydrase, this is the reaction that it is monitoring. As I told you, if you have it as a neutral pH, so generally your blood is at a neutral pH, right? pH of 7. Now if you have a neutral pH, as I said, all these reactions are spontaneous. So this reaction will proceed, but it will proceed with a rate of 10 power minus 1 per second. That means it will take around 100 seconds to hydrolyze 1 carbon dioxide, which is very slow for the amount of carbon dioxide that we inhale, right? But if this is at the pH 7. Now if you change it to a basic pH, right, because this is a nucleophilic substitution reaction. So the carbon dioxide is there, CO2. Sorry, nucleophilic addition. So this is how a nucleophilic addition reaction occurs there, right? So it is a very, very slow reaction in neutral pH. If you want to increase the reaction rate, so you need to make it a better nucleophile. You have to make the OH minus a better nucleophile, and it happens in basic pH. That means you can accelerate this reaction by changing the acidity of the reaction. That means if you increase the basic nature, if you increase to pH 9, pH 10 or something, this reaction will increase, the rate of the reaction will increase much faster, because you have OH minus will become a better nucleophile. But now in the body, in the blood, you cannot have like 7 for one reaction pH, 9 for one reaction, 4 for one reaction, and you know that in our stomach, right, it is highly acidic pH, it's close to pH 2 or something like that, right. So in our blood or something, you cannot have, keep on changing, you cannot have a pH for every different reactions. That is not possible at all, right, because then so this reaction is more faster in basic pH. There might be some reactions that are faster in acidic reaction, because in the same time in the blood there are multiple reactions happening. So you cannot change the pH of the blood for one particular reaction. That is not possible. That is not how a system evolves, right. Then when that reaction is going on, the other reaction has to stop, or that reaction will not proceed. So that means if you want to breathe, then you have to stop talking, right, or you have to, like, there are multiple reactions that are happening. So that doesn't happen. So it is possible to increase the rate of the reaction by changing the pH of the reaction. Now, how do the enzymes do that? It's a very interesting thing. So you can have a local structure, okay, where the enzymes can act, the substrates, in this case the substrate is what? Carbon dioxide. Carbon dioxide can bind the enzyme, and that local pH will be 9. Okay, so that means you don't have to change the pH of your entire blood or some entire system, but only a specific region. Once the substrate binds to this enzyme, there the pH is basic, and so in that case the reaction proceeds much faster. So let us see the example of this. So as I said, so it's not a carbonic anhydrase, I think I wrote it as 10 power 7, but it's close to 10 power 6, 10 power 7 depending on that. So the carbonic anhydrase increases the rate of reaction multiple times. So here I am giving you a simple table which we got from so this particular this thing, and it's also there in other. So I have given you only example of carbonic anhydrase, but for you to appreciate, just look at these enzymes, right? For example, there is AMP nucleosidase. AMP is adenosine monophosphate. You know, adenosine triphosphate, adenosine diphosphate. AMP is adenosine monophosphate. If you do the reaction in a test tube or in the lab, the rate of a reaction is 10 power minus 11 per second. That means for one reaction to happen, it will take around 69,000 years. But how much the enzyme does? Enzyme does the reaction in, it increases to 60 per second. That means it has increased the rate by 10 to the power of 12 times. I have given an example of 10 power 6 to 10 power 9, but here in this case you can see even the reactions that are much faster. So you see all these enzymes are doing nothing but your chemical reactions, but if you do this same reaction in the cell, in the test tube, it would take a long time, but the enzymes do it by increasing the rate of the reaction, and basically they do a huge rate enhancement, right? Now, as I said, how do they do that? For that, we will take the simple example of carbonic anhydrase. Although that is a very, very detailed structure of how enzyme catalyzes it. We are actually going to talk about enzyme kinetics little later. But for you to appreciate how they do it, we will just take the example of a simple carbonic anhydrase. Now, enzymes actually, so there are multiple mechanisms. We will come to that. But okay, so let us simply take the example of, so this is the carbonic anhydrase structure. Okay, I told you, right, what is this? This is nothing but you have a polypeptides that fold into a secondary structure, that further fold into tertiary structure, and then they kind of form into quaternary structure and make it into a multimeric structure or quaternary structure. So this is the structure of a carbonic anhydrase. So in this carbonic anhydrase, there is a region called active site. Active site means where the reaction is actually happening, right? So in this, so you see this is the active site. I have zoomed in. This is all done using, I think the pictures are from Wikipedia, and some of the, I have taken the originals from PDBID, which is protein data bank. You see, so that means if this is the active site, which is full of this amino acid called histidine. Okay, which is histidine. You must have heard of 20 different amino acids. One of the amino acid is histidine, which is very, very basic amino acid. So this is the entire enzyme, and in that there is a small region called active site, and that active site is full of histidines. Now this there is a cofactor called zinc ion. The zinc ion sits there, and then it kinds of this region now binds to the carbon dioxide, right? So the carbon dioxide, originally it was in a pH 7, right, when it was in the blood, but once it goes and sits in this active site is surrounded by a very large number of histidines, basic amino acids. So only this particular region will be very, very basic. So there is water around it, okay, and this particular region, active site, is very, very basic because of the large number of histidines. So once you have a basic nature in that, then the same reaction that I said, so you have histidines which is bound to zinc, which is a cofactor, to which more nucleophilic water is there, and then it will bind to carbon dioxide, and then OH minus becomes a better nucleophile and goes and attacks to that, and then the reaction is catalyzed. So I'm not talking about the entire mechanism, but I'm trying to tell you that, so within this places, so there are these enzymes where you, in the pockets which are called active sites, you can fine tune the amino acids in such a way. You can have acidic amino acids, you can have basic amino acids, you will have neutral amino acids, you will have hydrophobic residues, you will have all kind of amino acids, right? So you can fine tune the enzyme, or not you, the enzyme fine tunes it so that the activity inside this is very, very basic. So the region in this is very basic. So the original reaction that would have taken 10 power minus 1 per second, that has a rate of 10 power minus 1 per second in pH 7, now because in this active site the pH is 9, that has enhanced by 10 power, that has the rate of the reaction enhanced to 10 power minus 7, 10 power 7 per second, or 10 power 6 per second. That means this is how the enzymes work. So enzymes are not, they create a local environment that are feasible for performing the reaction so that it decreases the activation energy barrier. Okay, so this is a simple example of carbonic anhydrase. Now let me just show you a textbook model which is called, so that is a simple example of enzyme kinetics. Now you, as I said, you have a large, suppose I'm just writing this enzyme as some kind of structure, okay. Now if you have a substrate, okay, there are two more, so enzyme has to interact with the substrate, right. So if you have a substrate, either there is a substrate that exactly fits into this is called lock and key model, that means you have a lock and you have a key kind of thing that exactly goes and fits, or there is also called a new model, which is basically is what a lot of people are agreeing to. Suppose you have, this is a substrate, and so this will not exactly fit in here, right, this has a slightly different shape. Now this will reorient itself so that this can go and fit. So this is called induced fit model. So one is called lock and key model, the other is called induced fit model. In lock and key model, you already have a predefined enzyme that has an active site, and the substrate exactly matches with that, will come and bind to that. Whereas in induced fit model, the enzyme is very flexible around that active site, and depending on the only for a few substrates, it can modify, or you see, for example, this is how it was initially, right? It has changed the shape of it to fit that particular thing. This is called induced fit model, and a lot of enzymes are actually following this induced fit model. Okay, so this is just a cartoon representation, but I have shown you the structures of enzymes, right? So this is how enzymes create a local environment for the substrates, and then they do the reaction. Okay, so I guess you have understood and appreciated how enzymes work. Now shall we move on to the kinetics part of it? We'll do some mathematics and derive the expression of what I have shown you before. How many of you have done Michaelis-Menten kinetics before? Okay, quite a lot of you. Can you do it now? Okay, so let us start the reaction. So what is the reaction? You have enzyme that will react with substrate. It forms enzyme substrate active complex, right? Now, is this complex a covalent complex or a non-covalent complex? Is this a covalent or non-covalent? This is a non-covalent complex, as we said, as we showed. So most of the interactions have to be temporarily formed and they have to come down, right? So this is a non-covalent reaction. So a non-covalent reaction, most of it will always have an equilibrium. So enzyme plus substrate can form enzyme substrate complex, but this enzyme substrate complex can also dissociate back into enzyme and substrate, and this is where the enzyme interacts with the substrate and it will give product plus. Now this enzyme substrate complex to product, this product can also go back to enzyme substrate complex or not? Can this also be a backward reaction or not? Why is it? It is also an enzyme this is also non-covalent complex, right? So why can't it be a backward reaction? Energy of? So you have seen the carbonic anhydrous reaction, right? You have a zinc that is OH bound and then that is bound to your CO2 and then it is interacting. So your HCO3, if you see this product, right, this is your product, right? This, the one that is here, it's not visible. So this is the product, right? HCO3 minus, right? This can always be in equilibrium with this, no? Backwards. It is also a non-covalent complex. But then why don't we, but you are saying we can't write the backward reaction there? Substrate concentration is limited. What do you mean by substrate concentration is limited? No, you have enzyme, it binds to substrate. Enzyme converts the substrate into product, but it is still bound to enzyme, right? It is not completely released. After that it will release. There is a step where you have enzyme, there is a non-covalent substrate, and then enzyme will react with the substrate and convert, or basically the enzyme converts substrate into product. That is also non-covalently bound. So that can again, that enzyme, the product can always interact with the enzyme and form an EP complex or ES complex, right? So in principle, this is actually a possible reaction. The product coming, but in most of your textbooks, you will see that it is only one direction. You will never read about the backward reaction, but it doesn't mean that it doesn't happen. It happens, right? But this is called as approximation that we do, where we say that this is an equilibrium, this is only in one direction. Why do we do that equilibrium approximation? Is two reasons for that. One is, now you all know of Le Chatelier's principle. Now ES is in equilibrium with P plus E, right? Now if you do to start with your product is very low, or to start with your product is almost negligible, right? It is not there to start with, right? So if this is very less, the reaction is always in this direction, right? So if this is very less, or if this is negligible, the reaction always, even if there is equilibrium, the reaction always proceeds in the forward direction, because this is very low, right? So as long as the product concentration is low, you can ignore the backward reaction, right? And the other thing is, once the product is formed, unlike substrate, product might not have a specific interaction with the enzyme. So once the product is formed, this will actually leave the active site, right? The product will leave the active site. So always the product concentration is always low, because as soon as the product is formed in the enzymatic reaction, so now you have enzyme, okay, you have a specific substrate that is binding to this, right? Now the enzyme converts the product into some substrate which is of a different shape. This may not actually even stay here, because the enzymes are very specific to the substrates. So this product is not specific, so this will leave. So as soon as this leaves, the amount of product, the local concentration of product is always low, right? So that means this is always low. So if this is always low, the reaction always proceeds in this direction. The backward direction is neglected or negligible, but it doesn't mean that this doesn't have. So if you see any of the enzymatic reaction, we always determine what is called as initial rate, not the overall rate of the enzyme kinetics, but we only determine what is called initial rate. Initial rate means the time at which basically the product concentration is very minimal, that the backward reaction is not possible. Okay, so you always ignore this backward reaction and then do that, right? So this is your reaction, E plus S gives ES. Now I am just writing the rate constant for this. Let us assume that this is K1, the backward is K minus 1, this is K2. Now this K1 is, so have you heard of diffusion controlled reactions and activation controlled reactions? Diffusion controlled reactions means where the reaction rate is controlled by how fast the enzyme substrate complex, because the substrate, you have enzyme, you have substrate. So this is not just for enzyme kinetics, for any chemical kinetics or anything, there are two different kinds of reaction, diffusion controlled or activation controlled. Diffusion controlled means it depends, so the substrates are actually in the solution, they are diffusing across, right? It has to find the right partner, right? So it will, if there is more substrate concentration, if the molecule diffuses very fast, then it can find the substrate enzyme very fast, right? So those are, this is, so this particular thing is determined, is actually in diffusion control, whereas ES forming the product is actually activation control, because this is where the actual reaction is happening. The actual reaction of conversion of ES to P is happening here. So this will be diffusion control, this will be activation control. Okay, so E plus S is in equilibrium with K ES, which is in one way to P plus E. So this is the expression. Now let us derive a basic kinetics of this expression. Now how do you determine the rate of this reaction? So if I have to do a, so the rate of reaction is what? Rate of disappearance of substrate or rate of formation of product, right? So if I take a rate of disappearance of substrate minus ds by dt, what will it be? From this expression, what will be minus ds by dt? K1, okay, by a minus K minus 1 K concentration of ES, right? So this is the enzyme, this is the rate of formation of this. Now if you see, the enzyme concentration E is going to remain constant across this, but then this E will be different, so this total E will be summation of this. So if you want to take the initial enzyme concentration, E0 is equals to enzyme concentration at any time plus ES, right? So that will be your E0, right, to start with. Now, or you can also calculate the rate of formation of product. What is rate of formation of product? K2 into concentration of. So if you want to determine the rate of this reaction, this will be, here you have more terms, it will be easier to determine this, right? Because here E is changing, S is changing, but if you are keeping it as initial rate, this will remain constant, but this is always going to change. So K2 into ES. So if you can, but this is an intermediate, right? You cannot have an intermediate in the rate of reaction. So dP by dt is K2 into ES. So we need to determine the ES value in terms of initial concentrations. So only thing that we know remains constant is concentration of E and concentration of S. So we have to determine the concentration of ES in terms of E naught or concentration of S. Okay. Now some of you have done that. So how do I approach? How do you determine the concentration of ES? Steady state approximation. Okay. What is steady state approximation? Tanir ka? What is steady state approximation? Rate of formation of what? Product. So that means steady state approximation is done for product. rate of formation of intermediate is equal to rate of formation of or decomposition or rate of disappearance. So, rate of formation of intermediate is equals to rate of its disappearance, or if you want to put it as this, simply you can say that the change in the concentration of this is equals to 0. The change in concentration of your enzyme substrate complex or the change in the concentration of your intermediate is 0, right? Now, can you apply steady state approximation to anything? So, if I have A goes to B goes to C, K1, K2, can I apply, so where do I apply steady state approximation? I apply steady state approximation to B because this is the intermediate, right? So, can I apply steady state approximation to anything, or is there any condition? Can I apply, so steady state approximation means dB by dt is equals to 0, right, in this case. What are those reactions called? By the way, these are all called what kind of reactions? Consecutive reactions, okay. Okay, so when can I apply that steady state approximation? When K2 is much greater than K1. When K2 is when the rate of when K2 rate constants, right? When K2 is much greater than K1, only then you can say that dB by dt is equals to 0. But that is only if this case. But here we never said K2 is greater than K1 or anything. Now we don't know whether it is a diffusion control or whether it is activation control, we really don't know, right? But because there is an equilibrium reaction backward, so this concentration is always going to be remaining constant. So this will either go here or here. So this is going to be remaining constant. So dEs by dt is equals to 0. Now can you, do you know how to apply steady state approximation? Can you calculate the concentration? So what I want is to determine the Es, right? I want Es in terms of E0 and S, because this is a reaction initial rate. As I said, what we are doing is only initial rate. So for the initial rate, you can say that substrate initial concentration is S0. Right? To start with the initial concentration, we have a high excess of substrate and a very little amount of product. So your S is equals to S0. Now can you determine this Es in terms of this so that I can substitute there? You guys know how to do steady state approximation, right? Yeah. Can you do it in 2 minutes?
SwayamPrabha, Digital India, Educated India.